Here we are in the lab, and we're going to go through how everything's set up to do the DNA extraction. So there's one of the benches in the middle. White paper there is the revised protocol, so you need to have a copy of this in front of you as I go through the procedure. So there's all those materials. Structure bench at front. The important thing there is there's MiraCloth there, which we'll talk about in a bit. On the side back there, there's 2 centrifuges. And over to the left is a box of cheesecloth. All the material for the Kelex steps are along the back. And then over in the fridge freezer, so I walk over here in the refrigerator itself, we've got racks for the 2 tubes that you're going to save. Up in the freezer itself, we have frozen strawberries. So during the lab, each group's going to extract whatever fruit tissue it brought in, but part of one group should also extract strawberry because we know strawberry works really well and makes a lot of DNA. So you should wind up doing 7 samples in the lab, 6 of stuff your group's brought in and 1 strawberry for the 7th sample. Also in here, in the door and in the freezer itself, is the cold ethanol. So let's go back to the student bench here. What I'm going to do is pick up the protocol. I'm not going to read every word for each step, but I'm going to sort of walk through where all the materials are for this. So you brought in your frozen cup of fruit. You have a cutting board and a knife. You're going to check with the instructor about how to prepare your tissue. For example, if you've got strawberries, you're not going to use the pith, you're not going to use the leaves, just the strawberry itself. You're going to cut up your frozen materials. You have about 50 grams. So we have a weigh boat that's labeled for that, and of course the balance. You're going to take your tissue then and put it in the blender. Add in extraction buffer, that's the sort of bluish buffer over there, everything's labeled, and blend in the blender. And then you're going to get some cheesecloth again in here. Cheesecloth is back on the side bench. You're going to use the large beaker and the large funnel. Filter your extract through cheesecloth as directed, so you'll wind up with a large beaker. And then into that large beaker, after you get your cell extract, you're going to use a little bit of the protease, the meat tenderizer, which is down here in a bottle. Again, there's a weigh boat in here to weigh out the right amount of meat tenderizer. You're going to stir that mixture with a wooden stick, which is hiding in here on the other side of the extraction buffer, and then incubate that solution at 50 degrees in the water bath. The water bath needs about 1 inch of water in it, so there's enough water to heat up your solution, but not enough water to float and tip over your beaker. After you're done with that, you're going to take your beaker out and let it sit on the bench and cool down. Again, follow the directions. And then you're going to get some ethanol, which is over there in the freezer. You can bring that back and set it in an ice bucket, so there should be ice in the ice bucket. And after your beaker's cooled down with the cell extract, you're going to pour ethanol into the beaker to get 2 layers, as the directions tell you about. And your DNA should dehydrate and come out into that ethanol layer. Then you're going to take the small beaker and the small funnel. You're going to get 1 piece of MiraCloth. So MiraCloth here in the picture is in the red tray at the front. The MiraCloth is a little expensive, so 1 piece per extract. And you're going to filter your ethanol solution through the MiraCloth, trapping the DNA on the MiraCloth. Once you've filtered your DNA there, then you're going to take your MiraCloth and lay it down onto a clean cutting board, and you're going to use the spatula that's in there to scrape some of that DNA off, lift some of that DNA off, and fill up the 15 ml tube that's in here, which of course you need to label, till it's a half full. If your extract doesn't provide enough material to fill it half full, don't worry, but don't fill the tube over half full. You can get rid of the mirror cloth at this stage with any of the leftover DNA. Because now we're up to the stage where we're going to process the 15 ml tube and then do the Chelex. So with the 15 ml tube, again, following the instructions, you're going to centrifuge this to form a pellet and be able to pour off any ethanol that's left. So depending on the centrifuge you have, there's notes here for what speed to run it at. Make sure your tubes are balanced. So you're going to run the tubes, pour off the supernatant. Again, following the instructions, you're going to add some fresh ethanol. Just sort of rinse your pellet, spin it again, pour it off. You're going to let that dry. So after centrifugation, you're going to have a 15 ml tube that has a pellet in it that's dried. You need to add TE to that pellet. So the TE is over here on the side. There's sterile pipettes, TE buffer, beaker actually to use pipettes in. So you're going to add 1 ml of TE to your 15 ml tube following the directions and sort of flick that 15 ml tube to get everything resuspended. So as the instructions say, there may be some material that doesn't resuspend. You're going to let that tube sit for a bit so anything that doesn't resuspend settles out. And then you're going to come over here and get a Chelex tube. So Chelex is little pellets, right? So if you look at one of these tubes, and I'm not sure if it's going to show up, but if you look in the bottom, yes, you can see it. There's little pellets in there. That's the Chelex we're talking about. So you're going to take a Chelex tube and you're going to use the pipetter here to transfer 150 microliters of your DNA solution from your 15 ml tube into a Chelex tube, which obviously you need the label. And then you're going to mix up that Chelex and DNA solution. Save the 15 ml tube. That Chelex tube, you're going to put a lid clamp on, which is over here. And then you're going to put it in a yellow floater, and you're going to use the forceps to put it into a boiling water bath. You're going to boil it as directed, take it back out using the forceps, take your tube out, remove the lid clamp, so now you've just got a tube that has boiled Chelex plus DNA in it. You're going to take that tube and go down here, and again, each side bench may be set up slightly differently. You're going to go down here to the microfuge, and again, following the instructions, you're going to spin that microtube as directed, right? There's a little note there. This is to pellet all the Chelex in the bottom. So after you've done that, you can take your tube out, try not to disturb it. You're going to get a fresh empty tube that's right here, label it up, and you're going to transfer supernatant from that spun tube using a plastic pipette again into the new tube. And you're going to discard the Chelex tubes. You're discarding the one with the little pellets in the bottom, but you're keeping that liquid you took off the top. So at that point, if you get back to the bench, you should have two tubes. You should have your 15 ml tube that you resuspended in a ml of DNA, and you should have that new tube you made that's got the solution that came off the top of the Chelex tube. Both of those tubes then are going to go back into the refrigerator until next week. So again, there's all the racks. They're all labeled by section. On your tube, you've got to have a good identifier for your group. Also, write your section number on the tubes just in case things get mixed up. So basically, that covers how to make the DNA in the lab. Of course, check with your instructor at any point for questions, etc., because they can help work you through that.